Doctors and scientists can use data about the kidney to make healthcare and research decisions. An example of this data would be measuring the concentration of ions in different parts of the kidney. In this image, you can see a blood vessel that's running alongside a kidney tubule. So let's say that we took some measurements and found there to be 1.8 grams per decimeters cubed of sodium ions in the blood before filtration, and 1.3 grams per decimeters cubed of sodium ions in the kidney tubule. Exam questions might ask you to interpret this kind of data, so what might you be asked to do with this in the exam? Compare the concentration of sodium ions in the blood and kidney filtrate. Filtrate is the liquid filtered out of the blood and into the kidney tubules. So in this table, we can see the amount of sodium ions in grams per decimeter cubed in the blood before filtration and in the filtrate in the kidney tubules. So step 1, we can compare the amounts with a simple subtraction. So nice and straightforward then, 1.8 minus 1.3, which equals 0.5 grams per decimeter cubed. What's the next step? Step 2, make a comparative statement. Okay, so now we know that there's a 0.5 grams per decimeter cubed difference. We're going to write out that there's 0.5 grams per decimeter cubed more sodium ions in the blood than in the filtrate. You need to be able to plot graphs with data from the kidney. So what might that look like in an exam question? Plot a graph of the concentration of sodium ions in different fluids. This table contains the same data as we used for the previous question, but with an added reading for urine at the bottom here. So step 1, choose a suitable graph. The type of fluid that's been measured is a discontinuous variable. The types of fluid are categories, they can't be put onto a continuous number scale. Therefore, the appropriate graph would be a bar graph. Okay, now we've chosen a suitable graph, what's the next step? Step 2, label your axes. So we have an x-axis which runs along the bottom here and a y-axis that goes up like this. Remember, y goes to the sky to help you remember which one's which. So the convention is that the column on the left goes on the x-axis and the column on the right goes on the y. So our x-axis will have the type of fluid and the y-axis will show the amount of sodium ions in grams per decimeter cubed. Step 2, label your axes. So the amount of sodium ions is going to go on the y-axis, and remember to put the units for this. And on the x, we're going to put the type of fluid. Remember, it's the convention that the column on the left goes on the x-axis. What do we do next? Step 3, label your bars. Writing out each type of fluid is going to take up quite a lot of space, so we can label them instead. We're going to label them A, B, and C. Let's pop this onto the graph. If you do this, make sure that you write a key so that anybody looking at the graph knows exactly which one matches up to the type of fluid. Step 4, make a scale. When you're making a scale for a graph, first you need to find the highest number in the data. In this case, it's 2.2 grams per decimeter cubed. Next, you need to figure out the intervals that you're going to use. On our graph, there are 15 small squares available on the y-axis. So if we start at 0 and make every 5 squares be 1 gram per decimeters cubed, the highest plottable point will be 3 grams per decimeters cubed, which works for our data. Make sure that you also write these tiny marker lines next to the number so there's no doubt which place on the graph the number refers to. This frequently leads to lost marks in the exam. Now let's move on to step 5. Step 5, plot the bars. When you're drawing a bar chart, the width of the bars doesn't really matter as long as it's consistent, centered, and that the gaps between the bars are also consistent. So there we go, we've managed to plot a graph using kidney data.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy to follow videos and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE Biology course. See you there!